Great. Hello. Thank you, Amber. Thanks to the organizers for having me, and I'm pleased to share our work today. Dr. Demarest shared these findings earlier today at the parent conference, so I'll be reiterating them again. So again, I'm Zach Grinspan. I'm a pediatric epilepsy doctor at Weill Cornell Medicine in New York. The work that I'm going to share with you is really funded by the family and the parent communities. The trial that I'll talk to you about was funded both by the STX-BP1 communities as well as the SLC6A1 communities. These results, we got medication from Horizon Therapeutics at no cost, but they didn't support the study itself. We have since gotten some additional resources for them, including both funding as well as more drugs. So we're going to continue this work, which is exciting. So the study I'm going to talk about is phenylbutyrate for these two disorders. We had aimed to enroll 20, which we've done. All 20 have gotten their one-year outcomes, but I won't be able to talk. I think I'm going to present 6 to 10-week outcomes. And then a huge shout out to my collaborators at Colorado, many of whom are here. It's been like amazing collaborating with Dr. Demarest and his team. And then here are my collaborators at Cornell, also amazing people. So I'm going to tell you what you already know, which is SLC6A1 is 1 in 35,000. People with this disorder have developmental delay, hypotonia, intellectual disability, seizures, movement disorder, autism, attention disorders, and as has already been described, to disorder of the GABA transporter, which is this coffee bean in this cartoon. The other disorder that we did the trial on is STX-BP1 encephalopathy, similar incidence, about 1 in 30,000. We think epilepsy is maybe more common in that disorder. They also have intellectual disability, somewhat more severe to profound. Autism is common, hypotonia, movement disorders. This disorder is this blue peanut here. It's part of the snare complex, which helps synaptic vesicles bind with the cell membrane to release their contents. So there were two papers that led to this work, one by Dr. Jackie Buret at Cornell, and then the other by Katty Kang, who's here. I know Dr. Buret's work better, as the story goes, and some of you have heard the story. I met her shortly after arriving at Cornell as junior faculty. She described this work that she was doing about chemical chaperones. I did not really understand it, but I said to her, listen, if whatever you're doing works, by all means, let me know so we can try it. And a few years later, she found me, she emailed me, she's like, I published my paper, it works. I'm like, that's very exciting. Please come by. And we sat down in my office, we looked through her paper, and we Googled the different drugs that she had tried and then settled on phenylbutyrate. And then after we launched the study, Amber and actually Dr. Demarest approached me to say, hey, we're finding something similar with SLC6A1. Can we do this disorder too? So we ended up launching a trial with both disorders. The drug itself, for phenylbutyrate, it's FDA approved for urea cycle disorders. So in essence, this is a repurposing drug project. As I've been to several rare disease organization talks, this repurposing idea is a theme that many organizations are trying to pursue. Is there some drug that is already approved for something else that we can use for our disorder? So in this case, that's what this is. What's nice about 4-phenylbutyrate is it crosses the blood-brain barrier. It comes in two flavors. There's the sodium phenylbutyrate flavor, which is gross. It's salty. It's bitter. It's hard to take. And children with neurodevelopmental disabilities have a real tough time taking this unless they have a G-tube, and then you don't have to worry about it. Glycerol phenylbutyrate, on the other hand, we really haven't had a tolerability problem. It's a liquid. The kids, I think I have one family where the child calls it like his gross medicine or something, but like he takes it. So we're able to get this drug in fairly easily. I don't know how it works. I wish that I did. Every time I talk to a basic scientist, they tell me something different. these are the 5 stories that all seem to be consistent, and I don't know which story is right. Dr. Kang is nodding at me, so it may be that she's likely to have a strong opinion about which of these stories is correct. But I am a trialist. I'm a data guy, and I just work here. So here are the stories. One is it's improves trafficking. It gets the folded proteins to where they're supposed to go, or maybe the misfolded proteins to where they're supposed to go better. The other idea is maybe there's aggregates. There's like clumps of misfolded proteins that have some toxic effect on the cell, and phenylbutyrate somehow ameliorates that. The third idea is that it actually makes the proteins work better. That was, at least with Dr. Bure's lab, that was the core idea, this chemical chaperone idea that somehow phenylbutyrate kind of helps support the molecule and makes it work better. And that maybe applies to the mutant or the wild type, the normal protein. The fourth idea is this reduction of endoplasmic reticulum stress. If you have a lot of unfolded proteins around, that's toxic in and of itself to the cell, that there's this unfolded protein response, and that maybe it sort of tamps that down. And then the fifth idea is this histone deacetylase inhibitor idea, the idea being that somehow in facilitating or unfacilitating the rolling and unrolling of the chromatin, that it makes more protein expressed from the DNA. I was sufficiently convinced, maybe a little naive, because I hadn't done trials before, but was more than happy to give this a shot for these 2 disorders. So we launched a trial. I haven't said this yet, but I'll say this now. The parent-led organizations, and Amber, you deserve a huge shout-out for this. Just give a round of applause. They sure know how to light a fire. There was I put together a clinical trial design as best I could. We submitted for an initial round of funding and did not get it. But it was actually parents. There was a parent from, it was John Oldenhoff from STXBP1. He called me, and it was like Thanksgiving or Christmas, and he's like, Zach, can we talk about your trial? And I'm like, okay. And he went through it. He spent like 90 minutes with me on the phone. His professional job is designing phase 1 studies. So he was able to be like, you have to add this, and here's some language, and put this in there. So it was really with the support of the families that I, as the scientist, was able to put together a study that was able to get funded, and that we were able to get through the IRB and actually get it launched. This was the inclusion-exclusion criteria. For STXBP1, we were hearing that seizures were really, really core to that disorder, and so we required seizures for that disorder. With SLC6A1, and I've heard this from some of the parent talks and from Dr. Goodspeed's talk, seizures are kind of part of it. There's a lot of other stuff going on, and so it was clearly important to the SLC6A1 community that we include both children with and without seizures, which we did. There were a variety of exclusion criteria, none of which came to bear. What's nice about phenylbutyrate is that, other than the really gross taste of the sodium flavor, this is a fairly well-tolerated drug. These are the known side effects, and the ones that are highlighted in red are the ones we actually saw in the trial. Decreased appetite, there's a sweet body odor, metabolic acidosis. That did lead to one child dropping out, though we have since re-enrolled that trial because the seizures came back. And sleepiness, somnolence. What we find, just as an aside, is that those side effects happen when we're at the highest dose. For many children, we backed the dose off by about a third. The side effects dissipated, and then we slowly worked back up again, and the side effects didn't come back. So that's been nice to experience as a clinician. These are the outcomes that we've focused on: seizure burden, quality of life, communication, sleep. And I have some data on those. Development and EEG abnormalities we're still working on. There's two things, though, that I wanted to pause on this slide that we hear from families. Behavior is a big problem in this and other, DEEs. We didn't really capture it right in this study, though we're hoping to do better with subsequent studies. The tool that we're thinking to use is the aberrant behavior checklist, and I'm actually keen for feedback to know how well that captures. The other thing is movement, and Dr. Goodspeed, I was very moved by your slides, sort of thinking about that. We're like, yeah, we'll video the kids, and we were going to use the Sarah scale, and we stopped because the Sarah scale is not helpful. And then the videos were of variable quality. We weren't really sure what we were measuring, and so we have the movement disorder we've essentially stopped measuring, although we're still working on behavior. This is the study design that we have so far. We enrolled the children. We followed them with every week for 4 weeks, phone calls to see how they were doing. And then we brought them in for a 3-day, 2-night video EEG admission at Cornell or in Colorado. They were then on the drug for 6 weeks. Dr. Demarest has made it abundantly clear that as we go forward, we're going to do 12 weeks to align with more standard FDA trials that assess anti-seizure activity. We then bring the children back for a second 1-night admission. We had originally planned to taper, but the majority of children in the study have opted to stay on, and so we arranged for an extended use arm, which I think all but 2 of the 20 have enrolled in. So here are some preliminary results. Safety and tolerability, we've had really nice luck. The odor, sleepiness, and appetite suppression are certainly there, though they've been well managed. As I said, this one child had metabolic acidosis, stopped eating, needed to be hospitalized. But that child has re-enrolled because of the seizure effect was so favorable. And then tolerability, really few to no missed doses. Here's one example of a child with STX and his seizure outcomes. He was having 1 or 2 seizures on many nights, often in clusters. When he traveled up to New York, he had a cluster of seizures that we saw clinically and on our EEG as we were kind of titrating up the phenylbutyrate. But once we got to that dose, he's had a dramatic improvement in his seizures with just the occasional breakthrough. The response in children with SLC6A1 has been more dramatic. This is a 5-year-old child. I call him our respondingest responder. He was having 6 or more seizures every day, brief absences, similar to what Dr. Goodspeed was talking about. And Dr. Demarest and I are never sure, is it a seizure, is it not a seizure? But clinically, this is what the family was describing, these brief staring spells with the eyes kind of rolling up a little bit. He had a reduction as the phenylbutyrate was titrated up and has since been seizure-free. I think it's either this child or a similar one where the child was essentially seizure-free for a year or 18 months, and then they came back. And so I talked to the mom, and what we realized is the child had grown. And so the weight-based dosing had decreased. And we then increased the dose to match the weight, and the seizures again went into remission. These are our overall results at 10 weeks. There's 11 children across the 20 that we're calling responders, 50% or more seizure reduction. This is open label, and so in order to go in this category, we also had to say plausibly attributable to the drug, where the clinical instinct was also there were a lot of seizures. We started the drug, and the seizures kind of went away. There were 3 children that we're calling indeterminate, were either they met our criteria, but we're not sure it was the drug because there was other stuff going on, or they didn't quite meet 50% seizure reduction. In the STX-BP1 group, there were 2 children who had epileptic spasms, and this did nothing for the epileptic spasms. So it just sort of was a take-home point for me that infantile spasms, infantile epileptic spasms, really is a different entity, needs to be treated with high-dose steroids and/or vigabatrin. And then in the SLC6A1 group, there were 4 that did not have seizures, as we had noted. These are the 10 SLC6A1. All 6 who had seizures met that 50% or more reduction. The non-seizure outcomes so far, we're not seeing much. This is 6 to 10 weeks. These are scores for sleep, communication, that's the ORCA, and quality of life. And we're not seeing much different at 6 to 10 weeks. But again, we've got those 1-year outcomes that we're still crunching. One thing that Dr. Goodspeed highlighted that I want to emphasize as well is that SLC6A1 has an EEG signal that I think is a great benefit to clinical trial design because it's visually apparent. It's not. We need to think about the right quantitative assay to measure it so we can give it a number, but you can see it. This is an example. This is that 5-year-old. For the EEGers in the room, you're seeing some high amplitude delta. When us EEGers get together, we're like, is this notch delta? Is it spike and wave? And we'll kind of quibble about like the language to use. But visually, it's these high amplitude signals that are likely to be easy to measure if we sort of figure out the right measurement. Different than Angelman's, though, it's intermittent. So in Angelman's, you've got tens of minutes of slow spike and wave. And my sense, clinically is that those children are sort of in this epileptic encephalopathy. With SLC6A1, it's 2 or 3 seconds, 4 or 5 seconds. This on the bottom is a compressed spectral array. Each of those vertical yellow spikes is one of these brief seizures, and this is 1 hour's worth of data. So this child was having dozens of brief absences an hour. And then what I've told colleagues is maybe the best day of my career. I came in. This is a child who came in who had finished the 6 weeks and was coming in for the follow-up EEG. Again, dozens per hour to none. And there was another child in the study who had a similar finding on the same day. I paused. I told my residents, I said, let's just honor this. It's not going to get better than this for me. This is the moment. So this was extremely promising. And, what's nice about this is that if you can put a measurement on this, it's just very clean and very compelling. So limitations, it's just open label. You hear the enthusiasm in my voice, that's the clinician coming through. The scientist in me is much more skeptical. The scientist in me knows that I'm biased. I'm a doctor, these are my patients. I want them to do better, and that's gonna affect how I think about this. So I can't take that away, and I just need to acknowledge it. And so as you think about my findings, recognize that I am a very biased reporter. For SLC6A1, again, as Dr. Goodspeed said, there's absence seizures. So, like on the EEG, you can count them, but when they're home, it's hard to get a proper measurement of them. And then in the STXBP1, many of those children are having tonic seizures, and you always sort of wonder, if the parents are reporting that they're better, is it because there are less of them, or they're just not looking as hard? Particularly tonic seizures happen at night, and we know that those can be difficult to count. I'm working on the data. We have the 1-year outcomes. We do have some new funding and supply of phenylbutyrate from Horizon. They got bought out by Amgen, so it's now them, and they've provided us with some, again, funding and medication. We are expanding our work to other diseases. STXBP1, SLC6A1, and SYNGAP, you can kind of bucket as synaptopathies, that they're all kind of disorders at the synapse. And so we've next moved to try this on SYNGAP children. We've also thought about Dravet, Angelman's, creatine transporter deficiencies, and others. And then those EEG, I keep pointing at you, Kim, I'm sorry. we're trying to figure out how to quantify those EEG changes. A minute, I have 1 minute. So lessons learned about the regulatory approach to repurposed drugs. If you want to do off-label use of a drug that you think as a physician is a good idea, you don't need anybody's approval. You just write the drug. And so off-label use in clinical care is physician's prerogative and not regulated. If you want to do off-label use for clinical trial, which is what we did, you need some kind of FDA sign-off through the investigational new drug application process. For us, we got an exemption because we used the drug in a population they had already approved, children, and we used the dosing within the range of what was already approved. So it was very fast. Off-label use of expensive drugs like this, Ravicti costs $800,000 a year. That requires, the bottleneck is insurance. It's not FDA. It's convincing people to pay for it. And then finally, the IND exemption is a fast turnaround. I'm over time. Our approach for Ravicti is what's called the compendium approach. And so there are 4 compendia listed here that if you share data with them, they will list the drug as off-label use, and insurance companies respect that. So that's the approach we're taking in this case. I will stop there and say thank you all for listening.